Hi, everybody. Today is May 8th, 2025, and this is Neuroscientists Talk Shop, UTSA's Neuroscience Research Podcast. With us today is Kevin Bieniek. I knew I was going to ruin it. I was going to say Bieniek, but...
No, it's Kevin Bieniek.
Bieniek. Thank you.
Yeah, absolutely.
I'm going to just ask all guests to pronounce their own names, because I do such a bad job. Who's Associate Professor in the Department of Pathology and Laboratory Medicine, Director of the Brain Bank at the Glenn Biggs Institute for Alzheimer's and Neurodegenerative Diseases at UT Health, our sister medical school here in San Antonio. So Kevin's laboratory works on neuropathology of traumatic brain injury and neurodegenerative diseases, and he runs a brain bank that collects and analyzes brains of humans both with and without these kinds of brain injuries, and he works to harmonize the clinical phenotype and pathological diagnosis of people with brain diseases, and also maybe to understand that pathology, maybe that...
Yeah, absolutely. The relationship between what we see under the microscope pathologically and the disease that we see, clinicians and care providers see in the clinic.
Yeah. Great. I hope that's what we're going to talk about. So this is one of those things that famously sounds like it ought to work always, but in practice only works part of the time. And so one of the things he's doing is trying to improve methodology used by neuropathologists to better refine that side of the problem. So hi, Kevin.
Hey, it's a pleasure to be here. Thank you so much for having me.
Also with us, Melanie Carless from the Neuroscience, Developmental, and Regenerative Biology Department here at UTSA. And Melanie's familiar to our podcast listeners.
Hi, it's good to be here again, Charlie.
I'm Charlie Wilson. So, Kevin, there are a lot of interesting things we could talk about, but because we're in Texas, let's talk about football.
Yeah.
So, among other things, you have done a thorough study of a particular kind of pathology associated with, and I'm going to try to say this, chronic traumatic encephalopathy.
Yeah, nailed it.
I got it. Which results from repeated concussion like that suffered by football players and boxers, and maybe soldiers in combat as well, and maybe others too. We don't necessarily know all the sources of this. And could you start with just a little introduction to this particular kind of brain pathology and how you detect it, and what do we know about its causes?
Yeah, no, I think where I've been interested in repetitive TBI and sports and the intersection with brain and brain pathology is you typically don't think of neuropathology kind of very focused, looking at the brain at the time of death with epidemiology and kind of the different determinants of human health and disease and all of the kind of environmental exposome factors that we experience throughout our lifetimes. But certainly sports of all different types and at all different levels are very common in our society. And when I was first getting into this field, there were reports of CTE that were coming out in professional football players, NFL players, high-profile cases that were getting publicity on the news and, bringing a lot of attention into really kind of a burgeoning field at the time. But, a very basic question that always was kind of, personally motivating to me as somebody who played football and had family members that engaged in it too was, what is the risk of a disease like this to somebody who's not playing at these upper echelons of contact sports? So what about the youth and amateur athlete? And that is kind of what really led me, almost 10 years now, to get into this area and to explore how trauma at different levels and at different severities and intensities really kind of drives, neurodegeneration broadly.
So what does it look like? I mean, what is the behavioral phenotype, and then what is that brain phenotype?
Yeah, so CTE, chronic traumatic encephalopathy, is a neurodegenerative disorder. It's actually a pathology that we see and label in the brain. It's this protein called microtubule-associated protein tau. It's at the depths of folds in the brain called sulci, and it's around blood vessels. And it's kind of this constellation of pathology that pathologists can pick up and make as a diagnosis. But CTE is a biological disorder it's kind of been the nosologically used in the public lexicon to just describe a person with this disease as well. But the clinical diagnosis of CTE is called traumatic encephalopathy syndrome. And those that have traumatic encephalopathy syndrome, TES, with CTE pathology, we can see a variety of different presentations of this illness. So oftentimes in younger athletes with more mild pathology, you can see pretty pronounced behavioral changes in these people. Much more kind of severe rage and impulsivity. There can be very high incidences of psychiatric comorbidities. Whereas in somebody who is older with this pathology, you start to see much more of the classic, dementia phenotypes, memory loss, visual spatial impairment, executive dysfunction, and overlap with other neurodegenerative diseases like Parkinson's disease and ALS and Alzheimer's disease as well. So it can be really variable and it can be tricky to study, but it's certainly something that we see across a lot of different ages and exposures.
So is it progressive? I mean, if I get that when I'm young, is it just going to get worse and worse? Or if I quit getting hit in the head,
Yeah, no, that's a great question. That's still a question that's actually very actively being studied at the moment. The way that the gold standard for diagnosing CTE, more so than other neurodegenerative diseases too, is neuropathologically. But unfortunately, that means we have to get the brain at the time of death. When you get the brain at the time of death for a young athlete, or somebody in their early adulthood, it's hard to know how that disease would progress or maybe not, decades later in life. So now we are starting to develop better tools for diagnosing this early, and we're following people longitudinally in clinics to see how these diseases kind of dynamically progress, if they're static, if they regress, or if, they really kind of take off at a certain point, and what the modifiers are that really shape disease trajectories.
Because I could picture if I get some brain injury because of concussion, and then something has happened, some piece of the circuit's been disconnected or some elements have died, then I might just hope, okay, if I don't get any more concussions, I'm going to live without that piece of circuitry or those elements. But how would it get worse? But you mentioned tau, which is associated with progressive diseases.
Yeah.
So is it possible that some of the biochemical changes that you're seeing could somehow turn this into something that would spread on its own, like the seed of a neurodegenerative disease?
No, that's absolutely a great question. I think thinking of this disorder, tauopathies in general, they're a whole class of neurodegenerative disorders. They're typically advanced diseases in usually an elderly population. But this protein can be found in different cell types, in different locations of the brain. And then certainly a lot of the groundwork that's been laid in Alzheimer's disease, we do see that this pathology progressively spreads from one area of the brain to the next, possibly in a seeding or a prion-like progression strategy, transsynaptic spreading of tau. We still don't fully know that in CTE. There have been biological studies that do show that this protein can be transsynaptically propagated from one cell to the next. We're certainly, hoping to take some of the biospecimens and advance that research into model systems using organoids or using, animal models to study how these pathologies spread. Certainly for trauma, it's a little complex because trauma itself can be divided into different phases. There's a very short, brief acute phase of injury where there's actually damage to the neural system. You see axonal shearing, you can see diffuse axonal injury. And then there's this very pronounced secondary phase of injury where there's really, there's a lot of calcium and glutamate excitotoxicity. There's a lot of neuroinflammation. So all of these factors are kind of at play in a disease like CTE where there's really repetitive trauma. So it's kind of going through the cycle multiple times. And I think somewhere in that immediate injury, that neuroinflammation, this pathology starts to accumulate and build more in cells, and then likely progress to other areas of the brain the more trauma that occurs.
So I guess let me just state the question another way.
Oh, sure.
You
Absolutely.
answered it okay. It's just I'm not completely understanding what I'd like to understand about it. Like tau, many people who work on Alzheimer's disease consider tau a sort of causative factor. And why do I have Alzheimer's disease? Well, because you have a lot of tau. It's maybe that's an oversimplification, but it gets sort of said that way.
Yeah.
And in this case, do we think that you have brain injury after concussion because the concussion causes you to have tau, and then tau does the injury, or is it the other way around?
Yeah, so I think one of the things about thinking of tau and neurotoxicity is sometimes the tau that we see might not be the tau that's driving neurodegeneration. So typically when we're looking pathologically at a cell, we're seeing these large paired helical filament fibrils of tau, but there's a lot of suggestion that some are the smaller isoforms, some of these tau oligomers, might actually be more cytotoxic and might be more susceptible to spreading neuron to neuron than the big proteinous aggregates that we can visualize pathologically on the cell. So there's a lot of those studies that are still looking to profile tau subtypes within CT at the moment.
So can you see those little versions of it in your
typically,
immunocytochemistry?
There are ways to study oligomeric tau. There are specific antibodies to doing that. You do see those build up in these cells too. I haven't seen really conclusive studies that have shown oligomeric tau spreading from one region of the brain or in an area of the brain where we don't see aggregated tau.
Yeah.
But I think those studies would go a long way to kind of showing the progression of this pathology.
So in this particular kind of injury, the tau that you see is at the bottom of the skull?
Yeah, so where we're seeing pathology in these cases at the folds of the brain, at the depths of these sulci, around blood vessels, these are the areas where shearing forces moving through the brain are most likely to deform the brain and lead to breaking of the axonal processes, and I'm guessing an astrocytic end feed as well, more than likely.
So probably that just some mechanical shearing is what is the initial insult.
The initial force, and that's
shearing. Why it's happening right
Absolutely.
there. And then I guess just maybe I'm beating a dead horse, but if we imagine that that all the injury was limited to those places, since sulci are all over the place and on functionally different parts of the cortex, then the heterogeneity of the clinical manifestation would make a kind of sense. Maybe you've got sort of a lot of trouble with the sulci here and not there, and this is the place where you control your anger, and this is not. Is that going too far?
Yeah, it's hard to tell. I mean, I think a lot of times there's a lot of the tau is what, it's sometimes easier to see the gain of function than the loss of function in these diseases. It's easy to see the tau pathology build up, but I mean, also taken into this, there's severe disruption of the cerebrovascular unit or the neurovascular unit. There's a lot of white matter dysfunction. There's amyloid in diffuse axonal injury. APP can build up and accumulate. So I think there are, and you have this neuroinflammatory environment. We're actually in Alzheimer's disease, independently of this, have some indication that, microglia can actually propagate tau to some extent too. So I think in one way, with injury in minor stages of this disease, you see limited focal, what we call epicenters. And I think as disease progresses, the site itself can expand and become more dramatically impact in these areas. But at some point, there's a critical threshold where now all of a sudden, we're not just in this one site, but we're progressing to another area of the brain. And if that's being facilitated through glia, if that's transsynaptic spread, if that's blood-brain barrier disruption, it's not fully known and appreciated yet at this time, as much as I would like it to.
So white matter damage is like that because the axons are going from one place to another, and if you damage the white matter right here, you could be disabling neurons way
downstream,
far away. Lots of different kinds of things. So there's a lot of white matter sitting right at the place where you're seeing.
U fibers, deeper association, cortical tracts.
So can we look for what would we see in like a functional MRI of somebody who is suffering from this? Would we see changes in brain activity all over the place, or would they be restricted to the most damaged areas? Do we know that stuff?
That's a good question. It is known and studied. I'm not as familiar with functional MRI studies in CTE. I think part of the difficulty is diagnosing TES in clinic can be difficult because you have to rule out a lot of other things. It has to have a very specific syndrome. But there are studies that are looking at that. I think there's hypometabolism in the frontal cortex on like an FDG PET. I think the PET imaging studies are showing some of this pathology and dysfunction at the depths of sulci. And I think there is a lot more white matter hyperintensities. There are a lot of kind of structural MRI changes that we can see as well too. But my primary area has not been to focus on MRI imaging, unfortunately.
So one of the results that you have focused on is just how much experience with getting it on the head, I was saying, you need to have in order to develop this. And, we have our children are out there engaging in things that could potentially harm them. And that's a big issue that everybody cares about. On the other hand, we also think that these athletic activities are super important for their development in other ways. So what's going to happen?
Yeah, where's the balance? I think certainly being physically active in a critical developmental stage is important. I think as we study these sports, we can understand what types of contact sports or activities have higher risks, long-term chronic sequela, like neurodegeneration. And we're starting to see more strategies at mitigating incidental trauma in these sports. Things like concussion monitoring on the sidelines, things like rule and policy changes in a sport so a kickoff isn't as large or long. If we can do risk reduction techniques, I think that will make a meaningful difference. I think just monitoring, caring, sitting people after they experience concussions, I think that'll make a positive benefit. Perhaps some of the equipment upgrades. Hopefully, collectively, these things move the needle in the right direction. I think the downside, though, too, is these are sports in their very nature. There's always going to be contact. There's always going to be some degree or opportunity to experience a head injury in participating in these. So I think, if we can create alternatives like flag football on children, I think that is great. But there's always going to be this element or nature of the sport. And I think it comes down to, can we help protect our athletes? Can we monitor them if they experience this? And if, as we understand these diseases more, maybe there are potential interventions or prophylactic strategies we can jump in and kind of prevent this from progressing to a full neurodegenerative disease later in life.
We're not going to quit playing football. When I first came to UTSA, I moved from out of state, and we had a great dean here when I arrived, and Weldon Hammond. And one of the first things he did was to send me this email that said 10 things you ought to know about Texas. And they were really funny, and I wish I still had the email, but I remember 2 of them. And one of them was, the quality of a parking place is determined by shade, not by the distance to your destination. And the second one was, if you don't understand our fascination with high school football, you can just keep that to yourself. And I thought, these are 2 very true things about.
Yeah, some of the high school stadiums look like collegiate or professional.
We're not going to quit
stadiums
playing football. So what's the relationship between this kind of injury, which is pretty easy for us to understand how it comes about, and the kinds of injuries that happen in neurodegenerative disorders that may have a genetic component or arise because of environmental poisoning of some kind?
CTE has a very strong dose-response effect with neurotrauma, repetitive trauma, but even incidental traumatic brain injuries, a car accident, a fall, we can see that these sometimes will precipitate the emergence of a neurodegenerative disease, but they're risk factors for other neurodegenerative diseases as well: Alzheimer's disease, Parkinson's disease, ALS. So I think trauma can trigger other of these misfolded proteins to start to aggregate as well too. And we're interested in how TBI and Alzheimer's disease might be different from TBI and CTE.
So in your brain bank, you have all kinds of information about co-occurrence of these things, right?
Yeah, absolutely. And a lot of, as we age, there's higher frequencies of comorbidities or co-pathologies. So even somebody with CTE, there might still be Alzheimer's disease. There might be Lewy body disease as well. So we have a brain bank that really captures a wide variety of different neurodegenerative diseases. Certainly, with a research interest in TBI, we capture that information. But our goal is to deeply profile all of these cases to collect clinical data, to collect neuroimaging records and other biomarkers, so that when we get a brain and we can pathologically examine it for CTE or for another neurodegenerative disease, we can start to tie back some of this exposure data, some of the clinical picture, to understand the pathology that we're seeing under the scope.
This is a problem I think they all share, is that you're looking back at somebody's life after it's over, and you're asking, gee, did they get exposed to paraquat?
Yeah.
I have no idea. How could I possibly know? And so you search through where they've lived, and maybe you get some clues, but you never really have perfect data.
And I think there's always the opportunity to pair retrospective research with prospective research when we can identify candidates that we're interested in. Sometimes we have to get creative going to find the sources of information that give us exposure data, but we can take that information and then in turn kind of redesign a prospective study that would allow us to kind of capture that data and potential biomarkers and outcomes kind of moving forward too. And I think that's what we're doing with CTE now. We're actually designing studies of athletes who played at different times, maybe at the same school, and kind of following them prospectively and collecting this sort of data now.
I think that's one of the big things with any kind of repository like a brain bank is, as you start collecting things, you want to make sure you collect everything. So, it includes collecting the brain, but other tissues associated with it. I know, we've talked about the cerebrospinal fluid that you collect, also getting skin cells so we can then generate, these induced pluripotent stem cells and, model disease in a dish, essentially. But then there's also the data collection, do you have enough access to kind of medical records so that you understand the history of the individual, and then talking to family members to try and fill in some of those blanks and get that information as well. But one of the things I think with human research is, and this goes for both living and deceased individuals, is you rarely get all of the information. Yeah. You're either not told the information or it's just not available, or you don't ask the right questions. So that's right. How many times have you been hit in the head?
Yeah.
No, I don't know. A lot, probably, but
I am encouraged by new technology is kind of enabling some of this, though. I think of the smartwatch. Somebody who wears that might be wearing it to track their steps or just to see what time it is. But if we start collecting that data and aggregating it, there's potentially really powerful, findings that we can mine out of that and
ways in which to analyze that data to, see, is there something that looks like impact?
Yeah. I think for us, with a biorepository, we kind of have a twofold approach. We would really like people to come be seen in the clinic early in their disease and kind of get that full picture, follow them, do a really complete assessment of those cases. They have a ton of value when we get that brain tissue and we can look back again, comparing the tissue to their life, their story. There's still and always going to be cases, though, where we go into communities that might not have had access to care, that might not have the resources to kind of receive the same degree of care. And those cases are valuable, too, because they're understudied in brain banks that solely work with big clinics. I mean, we want to make sure that not only that we get these well-phenotyped cases, but that we get and study dementia that is relevant to our population.
So I was interested in what you said about pluripotent stem cells, and that's how long have people been collecting that kind of stuff in brain banks like yours? Do you have a huge collection of fibroblasts from all these patients?
Yeah, when we set up the brain bank in 2018, one of the things that, as Melanie had mentioned, we wanted to get the brain. We were also collecting spinal cord, pituitary, dura mater, stuff that's easy for us to kind of also build into our protocol, where we might not have an immediate use for it, but we try to have the foresight that this could be useful in research studies down the road. And so one of those things being a small dermal skin sample that we were isolating fibroblasts from alongside our partners at the UTSA Stem Cell Core. So we've banked a large number of cases, I believe 80 cases now plus, that have fibroblasts banked on these cases. We haven't converted all of these fibroblast lines to stem cells, but I think having the advantage of getting this brain and knowing at autopsy what the underlying diagnosis is, now we have an opportunity to come back to the fibroblasts to make a model system that we can study in vitro and really kind of dissect the biology underneath some of the pathology we're seeing.
Yeah, and I mean, that's the great thing is once you kind of start culturing these fibroblasts, if you treat them the right way, you can then freeze them, sit back, wait. In 10 years' time, maybe you get to use them. Maybe it'll be next year. But you have that opportunity to go back. And it's just that, again, thinking about that foresight when you start these repositories and what is everything that you could possibly imagine you could one day do with these samples and trying to prepare for that.
That's the whole idea of a brain bank, I guess. You don't expect people to do something good with every bit of brain you save, but you think maybe someday they will. So the nice thing about these being able to freeze these cells before you go to that intense labor making them into
it allows you to kind of do some things kind of on the cheaper side to get ready for, maybe one day you get funding to actually look at this in a big scale.
A giant
you have stuff waiting,
and you can cleverly select from it which ones to compare with which ones.
And it just becomes like a great resource at that point.
Yeah, and I've worked in the setting where within a biorepository you might not know the value of a sample at the time, but a new genetic discovery, a new pathology discovery can unlock clues in a sample that you have, and now all of a sudden you've collected that tool unbeknownst to you, which you can kind of go into this microphysiological system and really study it. And I think where I'm really excited by the organoids is the ability to really form very complex models, multicellular integrative models of the brain. Certainly working with brain tissue, I have an immense appreciation for how complex the histoarchitecture is of the brain. And organoids are a really cool way for us to study this in human-derived cells, modeling some of that same organization of the brain tissue.
And I think the good thing with organoids too is, there's so many different ways in which you can generate them, and, you can even sort of generate almost these subregions of brains, that sort of recapitulate, some of that developmental process and essentially the cellular patterning that you see. But you can do this for individuals that, have certain disorders, and you can kind of take a look and you can see differences in those organoids, potentially in the cell types that are there or how the organoid develops, sometimes how neurons might even migrate throughout that developmental process as well.
Could you give them a traumatic brain injury?
You can actually. It's interesting. Just last week we had a local kind of conference on epilepsy, and that's another disorder that is really well associated with traumatic brain injury. And we had someone give a talk there where they, in very simple terms, you shake a tube and you give the organoid a traumatic brain injury and see what happens. And they actually found like very high I guess electrophysiological changes, spikes that, you can't necessarily say it's a seizure, but it has that sort of profile that you see. So, that's certainly one area you can kind of think about as well is how good of a model it is. I don't know.
Yeah, that sounds
seems
great.
to show some things.
And opportunity to collaborate with people who are studying that, because I don't know about what you're used to, but groups of people like that suddenly get interested in your brain bank, not just
Oh no, absolutely. I mean, I think we can make observations in brain tissues, but we're always seeking for ways to kind of, again, understanding the biology, and to do that, team science is the best approach. So if you can partner with somebody who's looking at it from a different angle, who's modeling the pathology that you're seeing, or if you have a new target who can go into a system and intervene, try a new drug, look for a new biomarker. I mean, those are critical.
Wow.
We want a brain bank where we're not hoarding everything behind a refrigerator door. We want that material to be used. That's what our donors made their donation with the intention of, that their gift, their legacy is that they'll make an impact on scientific research and care. So we are, happy and encourage collaboration and sharing of this resource.
Well, great. Thank you very much, Kevin, for joining us.
Oh, thank you, Charlie, for having me.
And Melanie.
Yes, thank you.
And this has been Neuroscientists Talk Shop.